Welcome back. So this week we're going to start talking about more about DNA and transcription and translation, as well as turning on, turning off genes, using genes, not using genes, as well as how genes can be changed, mutations, and also biotechnology and how genetics has really changed the way that we live and the way that our societies have become more sustainable. So just as a review, remember we talk about genotype, which is actually what's in the genes, what the genes say, and then phenotype is actually what the trait is, what we actually see. Now remember, when we talk about how is it that DNA is going to code for something, well, we talk about DNA is going to create the mRNA, that is the transcription, and then the mRNA is going to code for a particular protein by telling the ribosomes what amino acids should come together, which is known as translation. Now, the genes in strands of DNA are a storehouse of information, and that is how that information gets relayed into mRNA, and then mRNA becomes the amino acids that are strung together to create polypeptides to create the proteins. Now, in transcription, the information goes from DNA to mRNA, and then in translation, we see that mRNA is going to code for what amino acids should be put together in order to build the protein. Now, we know that with DNA, A always goes to T, and T always goes to A, C always goes to G, and G always goes to C. But when we see mRNA, A is going to go to U, T is going to go to A, C goes to G, and G goes to C. And then once we have a code of our mRNA, let's say we have one that comes out like UAG, well, then we can take that and we can look up, hey, what amino acid is going to be taken to put together with another amino acid to form a protein? Which one is it going to be? We can just look it up in a chart because we always know, hey, UAG, well, let's find UAG. UAG right here, that is going to be a stop codon. All right, so now you see how you can create these proteins from DNA. It's not just DNA, boom, protein, but it's a set of steps with transcription and translation. Now, not all genes are going to be used at all times. Not all genes are never going to be used. There are ways that you can express a gene. You can turn a gene on, turn a gene off. So gene expression, when we use it as a term, it's the production of the protein that the gene sequence codes for. Gene regulation is whether a gene is turned on or turned off. It provides control whether the gene is currently producing the proteins that we want or it is not producing those proteins. And the environment often has this regulatory effect on gene expression. So gene expression is influenced by the environment. The environment has the ability to turn genes on and turn genes off. Now we talk about microarrays. Um, they are this powerful tool used to monitor the expression levels of thousands of genes all simultaneously in a lab. So particularly useful in exploring how gene expression differs in response to an illness or the treatment of an illness or aging or something going on in the environment. Now, cells have a wide variety of mechanisms for controlling gene expression, and we don't get into all of them, but it's important to understand that you have these transcription factors. So remember, we talk about transcription, turn that DNA into mRNA. Well, we have these transcription factors, which are these proteins that are going to bind to these special sites on the DNA, and they're either going to have a positive control, which initiates or speeds up gene expression, turns on that gene, or negative control, which is going to slow down or block a gene expression, turn that gene off. So gene regulation, extremely important during the initial development of an organism, fetal development. Um, you see that we have two different things that are going to regulate things. You can either have a promoter or you can have an operator. And a promoter is going to turn that gene on. So it's a region of the DNA that the RNA polymerase, all right, the enzyme that is responsible for making that mRNA recognizes and binds to in order to produce the mRNA transcript of the gene. So we're turning on that segment of the DNA in order to get mRNA from it. Or you can have an operator, which is a segment that is going to repress, is going to be a repressor protein, or it's going to be this molecule that represses and it binds to, and then it's going to ultimately block the RNA polymerase, that enzyme that is there for transcribing to the genes. So you have a promoter, you have an operator. Promoter is going to turn things on, operator is going to turn things off, and those are ways of changing gene expression. Now, just as an example, in the book, they talk about lactose. So you can have an operon that is off, where you have, if lactose is not present, that operon is going to be off, which is going to prevent the RNA polymerase from binding to the promoter. So now we can't transcribe that for that protein. You can't take that DNA, turn it into mRNA, and create that protein. But if you have lactose present, that operon is going to be on, and what's going to happen is that it's going to have this operator, and the operator is going to have the RNA polymerase then bind to the promoter and transcribe that gene necessary for lactose metabolism. So depending on if there's lactose in the environment, no lactose in the environment, that's going to influence if that lactose metabolism gene is going to be used or not be used. So there's many other ways that genes can be regulated besides operons, transcription regulation, activators, repressors, enhancer sequences, chemical interference. All of these things can play a part. Post-transcription regulation, but we like to talk about operons just as a general or a basic idea of talking about turning on genes, turning off genes, using genes, not using genes. So the gene is there regardless, but it's a matter of, are we using it? Are we not going to use it? Is that gene turned on? Is it turned off? So is it positive control, negative control? So environmental signals influence the expression, the turning on and the turning off of genes, as we like to say. By binding to DNA, regulatory proteins can block or facilitate the binding of the mRNA or the RNA polymerase and subsequent transcription of genes. Are you able to turn your DNA into mRNA and send it to the ribosomes to create that protein? So that is really what we're blocking. Regulation of gene expression also can occur in a variety of other ways that enhance or impede transcription or alter mRNA's longevity and rate of degradation, or influence translation or protein processing. So there's different ways to influence this, but we like to talk about operons just as a basic idea to understand how is it at all possible. We don't go into all the possibilities, but we talk about one of the possibilities and really what that looks like. Now, mutations are very important. Mutations are going to be an error in the DNA or a change in the DNA sequence. So this alteration to the DNA base, it may be detrimental, bad, or very rarely it can be beneficial. So certain ones are going to be beneficial, going to help us. We're going to see that it's going to help an organism adapt to the environment, be more fit for the environment, live longer, reproduce better, and they're going to pass on those genes. But a lot of times it's going to be a bad thing. There's also neutral mutations that have no positive or negative effect on the organism, and they can stick around for many generations, or they can kind of disappear. You see, like in fruit flies, you have normal fruit flies, which have these big eyes, and then you have mutant fruit flies, which have these mutated eyes. And it can change the protein produced by the altered gene shown here, where a mutation in a gene for an eye shape results in almost complete loss of the eye. So one little mutation, one mutation in the DNA can really change the whole trait that is seen. And mutations are actually a good thing. All right, they give us other options, and it leads to evolution. So mutations in non-sex cells like lung cells, not going to be passed on to the offspring. But if there's a mutation in the sex cells, no adverse health effects on individual carrying them, but they're going to pass on, or they're going to create offspring with those sex cells, and that genetic mutation is going to be passed on to the offspring. There's two major types of changes to DNA that may occur. You can have point mutations, or you can have chromosomal aberrations. We like to talk about point mutations because this is where we see just a little change in the DNA. So there's different types of point mutations where you're going to have a substitution where one letter is substituted for another letter. You can have an insertion where now a new letter is inserted and it kind of throws off the rest of the code. Or you can have a deletion where you're removing a letter and now, or nucleotide, I should say, and it is now throwing off the whole sequence of the rest. So there's different times, but all of them are going to really have changes on the traits that we see, or not all of them, but they all have the potential to change because if you change the genotype, then you can change the phenotype. And then you have chromosomal aberrations where you're going to see whole genes are going to be deleted. Whole genes can be relocated, whole genes can be duplicated. So in point mutations, one nucleotide is changed, but in chromosomal aberrations, you can see entire sections, right, of entire genes of a chromosome being altered. There's spontaneous mutations which are going to arise just by accident during DNA replication. Um, but you're also going to see that environment plays a role in this as well, where you have radiation-induced mutations such as skin cancer, chemical-induced mutations such as lung cancer. So the environment can play a role just as much as just random errors can play a role as well. You have faulty genes, which are going to be coding for faulty enzymes and can lead to sickness. Um, and then we talk about biotechnology. Now, biotechnology is really the mix of biology and technology, and we like to use this to our advantage, and it can improve food nutrition, make farming more efficient and eco-friendly. So genetic engineering is the manipulation of a species genome in ways that do not normally occur in nature. So we change the genetics, we change the DNA in order to make it code for something specific. Recombinant DNA technology has sped up this process, enabling the combination of DNA from two or more sources into a product. So you take a gene from another organism and you're going to insert it into the genome or into the DNA of another organism, and now it can code for that protein that that other organism could code for. Golden rice is a great example of biotechnology where to make golden rice, they took this beta carotene producing gene from other species. They introduced it into white rice, and that's how they made golden rice. And by doing so, it had it added vitamin A content to it. So now you have civilizations or people that are living in areas where they are vitamin A deficient. Now they can eat this golden rice instead of white rice, and they have this extra vitamin A in their diet. Almost everyone in the United States uses genetically modified crops regularly without knowing it. There's two factors driving adoption and modified crops in the United States. Plants with insecticides engineered into them, so you don't have to spray insecticides. They kind of make their own. Plants with herbicide resistant genes engineered into them, so they resist these these uh, um, uh, these herbicides that we grow on them, so there's not an issue when we spray them. And we're doing this so that we don't lose a lot of crops, so that the plants don't die off from these insects and these herbicides coming in contact with the plants. So we do this so that there's more output. So people argue that, is this exactly safe for us? Is it good for us? The verdict might still be out for that, but it sure definitely does uh, do a better job at feeding the world, or our growing population of over 7 billion people. It allows us to feed that many people and increases our carrying capacity by so much that you have to weigh the pros versus the cons. Now, when we talk about insect resistance, corn engineered to contain the spores of this BT or this bacterium called Bacillus thuringiensis kills insect pests but does not harm humans. So it has an insecticide in it. So what we do is we have these plants. Okay, they are destroyed by butterfly larvae, and we take this BT gene, all right, this gene that from this bacteria that is going to help kill insects, and we insert it into the DNA of a corn cell, and now we are able to breed these corn that are going to be resistant to insects. You have herbicide resistance as well, where they're going to be able to resist being sprayed with these strong herbicides. You want to have your plants be able to be sprayed so that it can kill all the weeds around it, but it won't kill the plant itself. Faster growth, bigger bodies. All right, you have larger salmon carries a growth hormone gene that keeps it growing year round rather than in the summer only. So now more food, more salmon when you're going to harvest those salmon. Rewards with risks. What are the possible risks of genetically modified foods? Well, featherless birds are cheaper for farmers and consumers, but there are unintended consequences, including vulnerability to mosquitoes and other parasites. So that is an issue. You can have, people argue that, well, if we have these herbicide resistant plants, we're spraying it with all these herbicides, the plants aren't dying, but then maybe aren't we ingesting those herbicides, and that is an issue. So while we have more plants and we can feed more people, what really are we feeding those people? So I don't necessarily have an answer for you, and you can go on and do your further research about this. But the verdict is out. This allows us to feed more people and keep people from starving around the world. So some concerns about genetically modified organisms. They say organisms that we want to kill may become invincible eventually. Organisms that we don't want to kill may be killed inadvertently. Genetically modified crops are not tested or regulated adequately. Yeah, eating genetically modified foods is dangerous. Maybe it causes autism. Maybe it causes cancer. Loss of genetic diversity among crop plants is also risky for the ecosystem. Hidden costs may reduce the financial advantages of genetically modified crops. So this is what people on the other side look at it and they say GMOs are bad, and the pros don't outweigh the cons. Not necessarily true. If you ask me, it's good. It's important to feed the world rather than make them starve because we don't want to try these new genetically modified organisms. But you can decide that on your own, and come up with your own idea about that. But there's always two sides, and it's not always clear cut that one is a better idea than the other one. So keep these things in mind as we move forward and we start to talk about evolution and where we see the genes and the genotype and the phenotype be a part of an entire system of different organisms.